Hello dear learners, in this video we have to learn about some of the important detection methods of those diseases which cause the mutation in the genes. So basically in our syllabus we have to study two important diseases which have been mentioned, one of them being the cystic fibrosis and the another one being sickle cell anemia. Now, both of the diseases are caused due to the mutations in their respective genes in their respective chromosomes. For example, the cystic fibrosis, the gene of the cystic fibrosis, that is the CFTR gene, it is present on chromosome number 7, and the gene for the sickle cell anemia is present on the chromosome number 11. So when there are certain mutations, which can be either point mutations or another kind of mutations, when these certain kind of mutations happen in these particular genes, in these particular chromosomes, then they lead to the diseases like the sickle cell anemia and the cystic fibrosis. So our aim is mainly to study about those techniques which can help to detect these mutations and which can help to detect whether the patient, a particular patient, either he is normal or he is a heterozygote to that particular disease, or he is a recessive one, means he is a homozygote to that disease. You should remember that both cystic fibrosis and sickle cell anemia are autosomal recessive diseases. So if the patient will have small a allele, then only he will be carrying that, he will be showing the phenotypes of the disease. If he is a heterozygote, this simply means that he is a carrier and he will pass on that particular disease to the next generation. And if he is capital A, that means he is perfectly normal and he does not have any symptoms of the disease in any of his genes. So the first technique which we are going to study today is the restriction fragment length polymorphism. So let me explain you the core idea of this restriction fragment length polymorphism. So basically RFLP analysis, what does it do? Let us take let's assume chromosome number 7, and let's assume that we are talking about cystic fibrosis. In a normal person, the gene for the cystic CFTR gene, basically it will be present in, obviously, in a pair. And suppose there is a mutated version of the gene which is present in another person who is carrying this disease or who is showing this disease. So he will have a mutation in that particular gene in that particular area, right? Now it can be either heterozygote when it is small a capital A, or it can be a perfect recessive homozygote in the case of small a small a. Now what does this RFLP do? RFLP basically is revolving around the core idea that there are the restriction sites present in the normal chromosome, and when there is the mutation in that normal chromosome due to certain conditions, these restriction sites get removed, and once the restriction sites are removed, the fragments would not be generated of the equal size. So it is always a comparative assay where we are trying to detect whether the mutation is present or not. So simple idea, if the mutation is present, the restriction sites will either be deleted, can either be added, or can be changed. As a result, if we are going to now add on the restriction enzymes, the fragments so generated would not be of the equal length as compared to that of a normal person. So that is what we have the core idea for the restriction fragment length polymorphism. So all those details here, again, they have been mentioned that the cystic fibrosis is the autosomal recessive disease, and it is caused due to the mutation in CFTR gene, and this gene is present on the chromosome number 7. Now this can be detected using the RFLP analysis, and this was one of the earliest molecular methods to detect the mutations. Now what happens here is when there is any kind of mutation, so this can create a new restriction site, or it can remove the existing restriction site, or it can alter the length of the DNA fragment. So suppose there is a normal DNA fragment like this, and now on this, suppose here we had the two restriction sites. In the mutated one, what will happen is that, suppose the mutated one has shortened. So let me shorten it a bit. Because of mutation, there were some of the nucleotides which were removed. So in that case, there would be one of the restriction sites present, but the another one might have been removed, or it might have been changed from its original location. So now what will happen is that once the restriction enzymes are acting on the particular sites, now what happens is that the DNA fragments which are generated here, they may be of these size. One is this, and one is this. But here it would be like this. So simply by comparing the fragments, we can detect that yes, the fragments are not of the ideal size, or the fragments are not of the identical size. There is some defect here, and that is why the fragments which are produced are different. So that is the core idea of the restriction fragment length polymorphism analysis. So how do we do it? First of all, we have to isolate the particular genomic DNA, and this genomic DNA we are isolating from the blood leukocytes. Why so? Because we have to detect the CFTR gene, and this CFTR gene is particularly very active in the blood leukocytes of the patient. So we are isolating the DNA from the blood leukocytes. Later on, what do we do? So yes, we do the restriction enzyme digestion. Now this restriction enzyme digestion can be done through PST1, EcoR1 or MST2. After that, we have to just analyze these DNA fragments on the gel electrophoresis, and we have to detect whether the mutation was present or not. Now supposedly, as compared to a normal sequence, suppose these kind of fragments are produced, and these are exactly equal fragments produced. Suppose like this way. Yes. So now what happens is that we say that the person is a normal person, neither is she a carrier nor is she infected with that particular disease. Now if suppose let's say the fragments generated one at one way are like this way and the other way are like this. So now we can say that here it was a normal chromosome, but here it was a recessive or a mutated chromosome. And suppose both of the chromosomes are like this only. So we can say that the person is definitely infected with that particular disease. So that is there if normal homozygous, normal size fragments, heterozygous carrier, both type of fragments in affected individuals will carry only the mutant fragments. Moving on further, we have to talk about the yes, these are the important points which you can by yourself also note. The core idea I've already explained. Moving on to sickle cell anemia. Now, sickle cell anemia, again, you have to remember the core idea. This whole disease revolves around chromosome number 11. On chromosome number 11, there is a gene, and the gene is called as HBB gene. Now, this HBB gene contains a kind of mutation which can lead to the sickle cell anemia disorder. Again, it is the autosomal recessive disorder. What happens is that in this gene of HBB, basically it has to synthesize a beta globin chain. Now, at the sixth position of the beta globin chain, there is a trinucleotide codon GAG, and due to a point mutation, always remember point mutation, it is replaced to or it is changed to GTG. So earlier it was coding for, let's say, glutamic acid, but now it is coding for what? It is coding for valine. So here the difference comes. So earlier, although it was coding for a glutamic acid, but now because it is coding for the valine, so this beta globin chain becomes defective one, and because now it is defective one, so the hemoglobin also will become defective, and because of that hemoglobin defectiveness, the RBCs will look like sickle shape. Now, one thing very important to note here is that once the mutation occurs, once the mutation is occurring, what happens is that suppose this is a normal chromosome number 11 and this is the normal, let's say, HBB gene. There is a site which is called as MST2 restriction site. In the mutated form, what happens is that this MST2 restriction site is removed. So now what will happen once you are doing the restriction fragment digestion, or once you are doing the digestion, so here the digestion will occur, but here no digestion occurs. So as a result, here we can get two strands, which should be actually the case, but here we are only getting one strand in the mutated form. So now this can be easily detected through the RFLP analysis, and we can say that yes, the person is either a carrier or is definitely infected from the disease, or is perfectly normal and is not affected from the disease. So here you can see I have also brought a diagram. So in the normal one, what happens is that there are certain MST restriction sites present. But in the mutated one, what happens is that one of the restriction site, means one location, maybe it has been destroyed. Now what will happen is a normal sequence can be produced here, but the disease sequence will be completely different, or the disease that fragments generate so would be of the different sizes. Why so? Because the restriction sites are, some of the restriction sites are missing. So in that condition, we can say that yes, the RFLP analysis has detected that there is the mutation in this particular person's chromosome. One core idea you need to know is that there is this southern blotting the technique which we commonly use in the RFLP analysis. It is nothing but transferring the DNA fragments from the agarose gel to the nitrocellulose membrane, and then from there we have to do the autoradiography to actually detect. Now, very important thing that you need to know is that in the sickle cell anemia, we can say that yes, generally what happens is that the MST2 restriction site is removed. But in the CFTR gene, when the mutation occurs, there is no such specific restriction site which is removed. So how do we detect? So what actually happens is that we will analyze the restriction sites present nearby to that fragment. Suppose here the mutation has occurred and here were suppose 4 restriction sites. Now the mutated fragment would be always be smaller. So although the 4 restriction sites are there, but the fragment size here would be higher and here it would be lower, or it would be small fragments produced. So we can easily detect that yes, because of the alternation of fragment sites, the mutation is present. So this is what I wanted to tell you. Okay, if the person is normal, the gene is HBA type. If there is a mutated gene, it is HBS. And here in the recessive homozygous form, the person will definitely be infected from the disease. Moving on to the next part, we have to now talk about the next important technique, and the next important technique is ASO dot blot method. ASO dot blot method is basically allele specific oligonucleotide dot blot method. The method is very simple, very easy to understand. It is just a kind of blotting only. So what happens is that here we have to prepare two kinds of oligonucleotides. The oligonucleotides are nothing but these are kind of probes which are radio labeled or they are fluorescently labeled. Now there are two kinds of probes which we have to first prepare before performing the experiment. One of them should be complementary to the mutated one, and suppose you are labeling it with green color. And the another one should be normal one, normal probe, and you are labeling it with the blue color. If you are not understanding what I'm trying to say, so let me explain you here. See, this is a normal fragment and this is a mutated fragment. Now see, if the normal fragment is containing this sequence, suppose, and the mutated fragment contains let's say AGACAG, like this. So we are preparing two different kind of probes. One is the normal blue colored probe which we are preparing for this, and this blue colored probe will contain the complementary sequence, that is TCAGTC. And one kind of probe which we are considering here is the one which will be complementary to the mutated sequence, that is TCTGTC. Okay, so what I'm trying to say is that we are using these two probes here. Now again, the same thing will be done. First of all, we have to isolate the DNA from the patient. Now after the isolation of the DNA, we have to first do the uh, blotting on the nylon filter paper. This can only be done that first you are doing the gel electrophoresis, after that you will do the blotting. So because of the blotting, what will happen is that your DNA will be transferred onto the nitrocellulose membrane. Now once the DNA is transferred onto the nitrocellulose membrane, you will bake that membrane at 80 degrees Celsius so that the DNA can be fixed. Also you need to do the prior treatments. For example, you have to treat with them with the other kind of things, for example proteinase K and all, so that the other non-essential proteins are removed. Once the DNA is fixed on the nitrocellulose membrane, now this DNA can either be normal DNA or it can be a mutated DNA. Now this DNA is fixed on the nylon filter, and the denaturation is also been done so that the DNA separates into single strands. Now we will add both the probes together on the nitrocellulose membrane. So we have added this blue probe also, we have added the green probe also. Now suppose if the DNA was normal, so the blue probe will bind to that normal DNA. And if the DNA was mutated, the green probe will bind to that mutated DNA. So either case can happen. So if the person is capital A, only the blue probes will bind. The green will remain free. If the person is capital A, some of the blue probes can bind and some of the other will be the green probes which have bound to the DNA which is stuck onto the nitrocellulose membrane. If the person is perfectly recessive homozygous, only the green probes will bind. Once the binding has taken place, now you will do the washing step. In the washing step, all the other unbound probes will be removed. Now you have to finally check the nitrocellulose membrane through the autoradiography or through enzyme linked whatever you do. So anything can be done to test what kind of probes are now attached to the membrane. Now suppose after washing you find that only blue probes are present on the membrane and all the green probes have been washed out. It simply means that your DNA was normal and your person was capital A, that is dominant homozygous. If after washing you find that both the kind of probes are still attached to the nitrocellulose membrane, it simply means that your person was heterozygous. If after the washing only green probes are remaining and none of them is blue, so it simply has detected the presence of that recessive homozygous disorder. So this is the core concept which we are using here in the ASO dot blot assay. So now we will be quickly taking a glimpse of whatever things we have studied through these texts. So yes, even in the CFTR1, what I was talking about, we know that the most common mutation is delta F508, which simply refers to that at the 508th position of the CFTR protein, your phenylalanine amino acid is missing. So it means that the trinucleotide which is coded for this phenylalanine is also missing. So here we say that there is a mutation, and now this mutation can be detected. How we will detect this mutation? You will take two kind of probes. One, the normal probe, which can bind to a normal DNA, and one will be the mutated probe, which will bind to that DNA where this trinucleotide is missing. And then you will again perform the ASO dot blot entire thing, and you will come to know that whether your patient is a normal person or a heterozygous carrier or affected one. Same we can do with the sickle cell anemia, and the entire theory remains very same, except for that this gene HBB gene is present on chromosome 11, and then there is a point mutation from GAG to GTG which codes for glutamic acid but now coding for valine, and the hemoglobin so formed is abnormal hemoglobin which can be designated by HBS. So the procedure remains the same. Coming on to the last method, that is the PCR oligonucleotide ligation assay. Now what do we do here? See, here also will contain or will consider two important cases. One case when the person is normal and one case when the person is recessive. So what do we do is we will prepare the probe for normal one and we will prepare the probe for the recessive one. For the normal one, we will prepare a probe which will bind to that sequence which is very specific to that sequence only. So one is the sequence specific probe and one is the adjacent probe. Here also one of them will be sequence specific probe and one of them will be the adjacent probe. Now what is the core idea here? Suppose we will take an example. If this is your normal fragment or normal DNA, and let's consider here to be a mutated DNA. Now in the normal DNA, suppose the sequence is ATGC, ATGC, and then the mutated DNA it is like ATCC, then again this ATGC whatever. So what do we do here is that we take a probe which is very complementary to this up till this part, for this also and for this also. So the two probes obviously they have to be different. One of the probe will be TACG while the another probe will be TACC. So this is the sequence specific probe for a normal one and this is the sequence specific probe for the mutated one. Now we have to take the adjacent probe which will be similar in both the cases. So here will be the adjacent probe and here also would be the adjacent probe. And what would it be? It would be TACG. Here also TACG. So please remember in the PCR OLA we take two probes. So what we are trying to do is that we are not taking a single large probe, we are taking two smaller probes instead. Now we will add DNA ligase to this probe. Okay, after this we have to add the enzyme which is DNA ligase. DNA ligase will act at this position. See, if the complete matching has been done of the probes and if the probes have completely aligned to their parental sequence, then the ligation will occur and the fragment or that oligonucleotide will be sealed. But suppose this is not the case and there is a mutation. For example, this was our parental probe, although it is mutated, but it is a parental DNA, not probe. So suppose here it was GG and the probe which you have added here has the A. So A is not complementary to G, so A will remain detached. Once this is detached, there cannot be ligation in any of the possible cases. So if there is no ligation, the probe will never be stable, will never be attached to that particular position. So in this way also, this is yet another way through which we can detect the particular mutation. So what it has been done here is that we have to synthesize a pair of oligonucleotide probe. Now the probe has to be in a way that there are two subparts of that probe, probe X and probe Y. Now what happens is that we have to take the normal DNA and then we have to attach this particular thing here. So one probe is attached here and another probe, adjacent probe is here. Also this B represents biotin, and this biotin is very essential for the probes to be separated later on. So biotin is an additional component which we are adding here, which helps in the detection and separation. So suppose it is a sequence specific probe and here it is adjacent probe. Now if the DNA was mutated, so although the adjacent probe is completely bound, but the sequence specific probe could not bind because here it was adenine, but here the mutation has caused it to be guanine. So in this case of mismatch, what will happen is that here the ligation could not occur. And once the ligation is not occurring, the probes will not bind. And if the probes are not binding, the colors cannot be detected. So we can say that there is a mutation. Okay. The another thing can be, once you are doing this, you have to add the DNA ligase. So because here complete sequence matching has happened, so the DNA ligase will just have to seal this nick. But here the sealing cannot occur because one of the nucleotide is still separated and it cannot be complementary. So in that condition, the ligation cannot occur. So this is the core idea here. And why PCR? Because whatever the gene of interest is, where we have created, we have to first amplify it. So once the amplification has been done, after that only we will be adding our particular probes to detect the mutation. So it is always a combination of PCR and oligonucleotide ligation assay. So again, it will be same repeated for the CFTR also, and then the same thing can be done for the sickle cell anemia also. The core principle and the core idea remains same for both of the diseases. So in this way, we have covered the entire 6 detection methods for the 2 diseases. So this covers our topic.